talk. So our presenter is Steve Gregorich, who is a very prolific researcher and author. I can read some of what we put on the announcement, but I can just tell you quickly that Steve graduated in with a degree, with a Ph.D. in quantitative psychology. So he has both psychology and statistics, I guess, from the University of Texas at Austin. He is a very prolific researcher. I checked, he has almost 200 publications, and quite a few of them in 2020. I checked his profile at UCSF. His network concepts, 553 items, co-authors 132, and the list goes on and on. Anyway, Steve and Tor and Chuck McCullough presented a few weeks ago a general presentation about research that is happening in these times of COVID, and what happens with projects that started before and were interrupted and are continuing. So this time, Steve is going to expand and make some corrections and have more time to cover things. So we are looking forward to it. Steve requested that you can put your questions in the chat box, but Steve prefers not to answer during the talk unless this is something that is not clear and you need to have the answer right away. Otherwise, he'll reply to the questions at the end of the presentation. And I'm really sorry we cannot take you out to lunch after the talk, Steve, which we used to do in normal times, but hopefully we'll do it in 2021. Okay.
Well, thank you so much, Esti, and thanks for you and Tor for hosting this and giving me more time to cover this topic. It's something that when Tor first contacted me about what to do statistically to deal with pandemic effects, you know, my first thought was probably not very much, but there's really a lot that you can do. So I got encouraged and interested in it. And, you know, I will say that this talk is trying to cover a lot of ground. There's a lot of different kinds of scenarios that I am considering. It's a lot. This talk is a beast. You're really gonna have to like hang in there with me. And I haven't really had a chance to practice it as much as I should have for a talk that's as complex as it is. So bear with me. And also, I mean, I don't wanna discourage questions during the talk. I've just asked SD to kind of moderate if a question could wait till the end, but if it's a burning question, then ask it and I'll try to address it. So can everyone see my slides? Okay. So the first couple slides are going to be the same as the last month's talk, but the whole idea is you've got a study that's been interrupted by pandemic onset, and you want to find out whether or not the pandemic will affect your study results. So you need to diagnose whether and how it might be affecting your results and see if you can accommodate it somehow. Oops. Got to get the right mouse going here. So there's going to be pandemic-related impacts on a study that are under investigator control. So it could be anything. You could abandon your study. You could completely change the aims. But more often than not, what's happening are modifications that might affect data collection or the way you interface participants, intervention delivery, and other kind of design feature changes. And then there's changes that are not under investigator control. So there's just going to be a lot of secular contextual factors that are happening. And some of those might affect the participants' relationship with the study. You might have different kinds of participants enrolling in the study in the pandemic era versus the pre-pandemic era, et cetera. Your participants could get sick with a pandemic disease, et cetera. So there's a lot going on. It's all happening at once. You've got study design changes, you've got potential participant reactions or different types of participants involved in your study. And so as the investigator, you're just wondering whether all of these pandemic-related changes could impact your study results. Before I move on, I just want to say that it's good to document as best as you can all of these changes, the changes that you made as an investigator, the changes that you know about, like policy changes that have happened in the, just secularly, shelter-in-place orders, things like that. If you know individual participants, pandemic-related factors, you might want to record those. Think about adding some variables to help with those kinds of things, because all of that is going to be useful for sensitivity analyses that you might conduct. I'm not going to talk about sensitivity analyses today, but just wanted to make a note of that. So you could, if you have a study that was ongoing at pandemic onset, so you've got some pre-pandemic data, some pandemic era data, you could conceive of three basic types of estimates. There's a pooled data estimate, where you basically put all the data together, including pre-pandemic and pandemic era, and estimate a model using all of the data. And hopefully in some circumstances, some of those model estimates will be deemed to be equivalent across or not modified by pandemic status. And so you would have access to your full data for those estimates. But in some cases, you may really end up needing to do something more along the lines of a stratified analysis. So you might have pre-pandemic estimates where you just kind of ignore all of your pandemic era data and just kind of fit your model to the pre-pandemic data, and the same thing in the reverse for the pandemic era estimate. But not all study designs are going to support all of these, at least not allow you to estimate all the effects that you would like for all three types of estimates. So this is a very simple example. If you had a cross-sectional study where half of the participants were recruited pre-pandemic and the other half after pandemic onset, you might have a goal of estimating, say, the association between dating app use and unprotected sex. So a diagnostic question is whether the association of your X and Y is roughly equivalent across the two pandemic periods. So along those lines, you could fit a little diagnostic test. You could test the effects of app use, pandemic period, and their interaction on the outcome. And if the interaction is significant, or I should probably say substantial, because you're not just going to be looking at p-values here, you should report effects stratified by pandemic period, because there's two different effects that are happening, and to pretend that they're the same may be misleading. You would come up with an app use effect estimate that doesn't represent either the pre-pandemic or the pandemic era period. But if there's no interaction effect between app use and pandemic period on the outcome, then essentially means that the relationship between app use and the outcome is equivalent across periods. And so you could then report a pooled data estimate, and you'd have access to all of your data and better efficiency, power, etc. So moving along to a longitudinal study, and say baseline 6 and 12-month outcomes that are going to be fixed, modeled as fixed times relative to enrollment in the study, you might have a situation where you've got these different sequences of pandemic status within certain subsamples of your overall sample. So the 25s mean 25% at each time point. So you've got this top row, which is, 25% of your sample had all of their observations pre-pandemic. And then the bottom row is 25% who are all enrolled and completed the study in the pandemic era. And then in the middle, you've got these kind of combos. The first folks finished 6 months before the pandemic onset happened, and their 12-month was during the pandemic era, and the opposite for the third row. So here, pandemic status and time are completely crossed with each other. They're not crossed and balanced, but they're completely crossed. So each pandemic status is observed at each time point. And in this case, you could fit a model to the pooled data and get some pooled data estimates, hopefully. Or you could just work with the pre-pandemic data for a stratified pre-pandemic analysis, or you could do a stratified analysis of the pandemic era data. So here's our same kind of setup, and I'm calling these are sequences of pandemic status. And then this particular layout shows 4 sequence sets. Each row in that graphic is a different sequence set, pandemic sequence set. And then you could have a time varying pandemic indicator for each observation. So for any one participant, for any assessment that either happened at the pandemic, pre-pandemic time, or after pandemic onset. So I think I've got these backwards. It should be 0s for the blue and 1s for the red, but that's okay. So you could, if you had a model like this, you could think about it. It could be a randomized controlled trial. It doesn't have to be. It could be an observational study where you've got a stochastic X variable that's your main predictor variable of interest. It could be either kind of study. But if we're thinking about an RCT, you could fit a diagnostic model that includes a group time pandemic main effects plus all interaction effects. It would include group by time. That's not listed there. All 2-way interactions and the 3-way interactions. And then you would perform some backward elimination to remove the non-substantial interaction effects that include pandemic. And I say non-substantial because depending on your power, you could have a non-significant interaction effect that's fairly substantial, or you could have a highly significant interaction effect that's not substantial if you had a huge sample. So use some judgment about how much effect modification is happening. And then so the impact, if you found a group by pandemic effect, that means that there's a different group main effect within each pandemic state. So you wouldn't really want to report the pooled data group main effects because they would neither reflect the pre-pandemic or pandemic eras. And same thing for time by pandemic. If there is a significant or substantial interaction effect there, then you don't have one set of time main effects. You've got a pre-pandemic set of time main effects and a pandemic era set. And the same thing for group by time. So if any of these are significant, then it means you really should be reporting separate estimates by pandemic era or pandemic period. But if you're thinking about on RCT, there's a lot of different ways you can report treatment effects. You can report the treatment effects by looking at group differences. You can report the treatment effects by looking at group by time interactions. So in some cases, maybe you'll find that the group by time by pandemic effect is not substantial, but the group by pandemic effect is. So it might be in your best interest then to focus on group by time interaction effects as your main outcome of interest. So there may be some changes happening to your analysis plan as a result of the pandemic, which is, I guess, is an understatement. So what I did is I conducted a bunch of simulations, and just explaining the scope of the intervention or the simulations is going to take a while, so bear with me. For each condition, I just generated one big sample of 10,000 participants, each assessed at 3 points in time. So there's 30,000 observations in each simulated condition. And so I assumed an RCT with 3 times that would be modeled as fixed times, 2 pandemic states. And so the pandemic states are going to be balanced across groups, across intervention groups, because you're moving along, you're randomizing people as you go along. And so whatever, you're always going to have both pandemic states observed in both intervention groups. And again, it doesn't have to be groups. It could be some X variable that you're measuring from participants, but even so, you would expect probably that the distribution of pandemic states would be, fairly equal at all values of that X variable. That could change if the pandemic really altered the types of people who are enrolling in your study, but probably it won't be a concern, at least under our current circumstances. I don't think it often would be. The simulations assumed 4 sequences of pandemic status, as I described before. And then there's these data generating models. So I chose 5 data generating models. And the first one is basically, it just has group by time, and pandemic main effects. And so I just wrote up a little equation here that explains it. And then this table just kind of, you don't have to remember any of this, but it just gives a sense. These black values are the population means. This is the control group at 3 times. This is the intervention group at 3 times. Then down here, these are the group differences at each point in time. So 7 minus 2 is 5. And then these are the simple time effects within the control group. So 2 minus 0 is 2, etc. And in the intervention group, and then we've got, so these are the differences, and these are the difference in differences. So this is basically the group by time interaction effect during the pre-pandemic period. So this is an overview of the 5 population models. So the first one just has group by time, and pandemic main effects. So this G bar T is SAS shorthand for, instead of writing group by time, you can just write G bar T, and then that encompasses both main effects and the interaction of all of the effects involved. So in this case, we've got group by time, and pandemic, and we expect that the pooled data estimates will be unbiased for all 3 group time and group by time effects. For population model 2, I added to population model 1, a group by pandemic effect. So now in this model, I'm going to expect that my group main effects aren't going to be equivalent across pandemic states anymore. And so reporting a kind of pooled data group main effect might not make a lot of sense. But my time main effects and my group by time effects should be okay to report as pooled data estimates. And then there's population model 3 that kind of replaces the group by pandemic with time by pandemic interaction. And you can see the expectations here in terms of what's going to be biased and not. Population model 4 includes all two-way interactions, and population model 5 includes everything up to the three-way interaction, where all of your group time and group by time effects would differ across pandemic states. And you could say they're biased in that sense. They're biased by pandemic status. So again, you don't need to look a lot at this, but the pre-pandemic expectations are going to be identical for all population models. That's this table. The pandemic era population parameters are going to differ by population model because of the different interaction effects that are included. So that just ends up with different expectations for what the outcome would be. But if you wanted to, you could go through this exercise of looking at this pre-pandemic table and getting the group by time interaction effect, say, in the pre-pandemic, and then this is the group by time interaction effect in the pandemic era, and then this is the difference between those. So this is the group by time by pandemic interaction effect in that particular circumstance. So still talking about simulation setup. So I started this talk with the 4 sequences. This is the upper left corner here. The full simulated data set for any condition includes these 4 sequences. And then, but that's not always going to happen, and that's a pretty good circumstance because each pandemic state is observed at each time point. But that's not always going to happen depending upon the timing of your study, when it started recruiting, how long recruitment is, how long it takes you, that is, how long you're following people up. You might find that your study falls into a different set of sequences. So what I'm calling S123 would include the first 3. So here there's no pandemic era baseline that's observed. And then 2, 3, 4 is kind of the opposite. There's no pre-pandemic 12-month assessment. Then I've got 1, 2 over here where the pandemic era is only observed at 12 months. And the opposite of that is 3, 4, where pre-pandemic only is observed at baseline. And then S2, 3, which is really the worst. If you just got rid of the 6-month assessment, then you would have baseline and 12-month follow-up completely confounded by pandemic status. So you're hanging on by a thread here by having at least both pandemic states observed at 6 months. So there's 5 population models. There's 6 pandemic status sequences that are possible. And that's 30 simulated conditions. I'm not going to go through all of them. I will try to see if it's worthwhile to go through all 5 population models for the sequence 1, 2, 3, 4. And then for the remaining sequences, I'll just report upon a model here and there. Additional simulations. There's a variation of population model 5 that has a simpler interaction effects between pandemic and time and pandemic and group and time that I'll discuss. And then I also want to mention that, especially for studies that don't include a pandemic era baseline, the investigator might want to consider adding a pandemic era baseline if it provides something of interest to them, and I will discuss that a little bit more. So this is S123 with this star star 25 star star cell. That's the augmentation. You know, how large that group would need to, or that sample would need to be, is a question for your statistician to figure out the kind of efficiency gains and the extra parameters that you'd be able to estimate with that. Okay, so we're done explaining the simulation setup. And so there's 4 types of models that you could fit to simulated data. The first is a diagnostic model. And so you're basically going to fit an Ancova model with up to the 3-way interaction of group, time, and pandemic, and do some backward elimination of non-substantial effects that include pandemic. And what you end up with is a final empirical model that I'll just label as the analysis model. So in this case, time has 3 levels, so it has 2 degrees of freedom. So this analysis could give me a 2-degree of freedom test of the time effect, the time by pandemic effect, and the group by time by pandemic effect. But if there's more than 1 degree of freedom on that test, then I might want to break down those effects to understand whether there's 2 degrees of freedom, say, worth of substantial interaction effect or not. So in addition to a basic model, I wrote some estimate statements to test. So this is basically, so there's an effect of time from baseline to 6 months. Is that modified by pandemic? And this is the same question, but for the 6 to 12-month transition, and the same question for the baseline to 12-month transition, ignoring 6 months. And you could do the same for whether or not pandemic modifies the group by time effect, and look at the simple effects there. Just diving in a little deeper, not wanting to only focus on the multi-degree of freedom omnibus tests. So the second type of model that you could fit is what I'm just calling an Ancova model. And it's just like the diagnostic model, except it's being fit to the analysis model, the one that you pared down possibly from the full model with up to 3-way interactions. One of the things I'm doing is I'm looking at 16 simple effects. So this is say an RCT with 2 groups, 2 pandemic states, and 3 time points. And so if I just think about all the different kinds of effects that people might report upon in a randomized controlled trial, a lot of times they're interested in group main effects at each assessment time. So treatment versus control at baseline, treatment versus control at 6 months. There's 3 of those. A group main effect average across 6 and 12 months, say the intervention was finished before the 6-month assessment, so both 6 and 12 months are follow-up outcomes. And so in some cases, you might want to average those values and compare those across groups. There's going to be 3 simple 2x2, 1 degree of freedom group by time interaction effects, all based upon the time span that they're focused on, say baseline to 6 months versus baseline to 12 months. It's going to be simple effects of group within each time. So baseline to 6 month difference in the treatment group. And then you can also consider group average effects of time. So first you're averaging across groups at baseline and then averaging across groups at 6 months and comparing those. Less common reported, but I thought I would include those anyway. So you can fit basically an Ancova model of your analysis model that you determine from your diagnostic model, and then have a bunch of custom estimate statements to produce your 16 simple effects. Then there's one I'm going to call an Ancova model. So I'm not trying to say that there's a difference between ANOVA and Ancova in this context, because you could get the same results either way. But in SAS, specifying this as an Ancova model where pandemic is treated as a, quote, continuous covariate is really going to simplify a lot of things just because of the commands that are available and what they can do. And essentially, in this model, pandemic is no longer included in the class statement, so it's considered a continuous covariate. And then you still specify your analysis model, whatever that was. And then instead of a bunch of estimate statements, you can have a bunch of what these are called least squares means estimates. Different way of obtaining custom estimates, but it has this feature where you can specify the covariate value that you want these weights applied. So I can create 16 LS mean estimate statements to obtain my 16 estimates of interest in the pre-pandemic period, and another 16 for the pandemic era. So it'd be like this one, where pandemic equals 1. Again, I could obtain these estimates in the ANOVA framework, but it'd be a lot more finicky. I'd have to like get my pencil out and really start thinking about especially when the cross tabulation of pandemic status and time is not fully crossed. You have to be careful about how you go about this. Okay, then the last type are stratified models. So all you're doing is for pre-pandemic stratified model, you just ignore all the pandemic era data and you fit your analysis, you fit your model to the pre-pandemic data. And then you can have your estimate statements for your 16 effects. And depending upon how much data are available at each time point in each pandemic state, you may not get a full set of estimates, obviously, but you can try, and it's a worthwhile thing to do under any circumstance, and we'll talk more about that. So stratified models are, I say they're safe harbors. If you can estimate a parameter in a stratified model, stratified by pandemic status, there's no concern that it's biased by pandemic status. You've either got a pre-pandemic or a pandemic era estimate. So there's no concerns about, whether or not it's a meaningful estimate. Okay, so I've gotten through the simulation setup. I've gotten through the different kinds of models. So in general, the Ancova approach is going to be equal to or better than the ANOVA approach. Again, you could make either one of those. You could make Ancova or ANOVA produce the same results, but just the way that they've been set up here, Ancova is going to be equal to or better than. Stratified analyses, again, it's a last resort, but they do have some diagnostic function. And so mostly what I'm going to do is present Ancova and stratified estimates side by side. I'm going to present ANOVA like once or twice. And then for jointly unbiased estimates, which means that, well, first of all, it's a simulation, so we know what the population parameters are. But if the Ancova result and the stratified results are equivalent, then that means they're unbiased. And so if both corresponding estimates are unbiased, then I'll also present a relative efficiency of those estimates, and that's calculated based upon the standard errors. So I get, just squared standard error for, say, a group effect at baseline in the stratified model over the squared standard error for the Ancova model. And that can be considered a kind of a sample size ratio. So essentially, if I got a relative efficiency of 2, it suggests that a stratified analysis would require a doubling of sample size to gain the same efficiency as the Ancova model with the original data. So that's important to consider because what we want is the most efficient, AKA most powerful analysis. So, okay, now we're getting into simulation results finally. Thank you for your patience. The first simulations consider this sequence 1, 2, 3, 4. So groups, again, always going to be fully crossed with pandemic status. But in a lot of studies, time will not be fully crossed with pandemic. But in this case, it is. So this is, this top graphic shows the 4 sequences that are included in the data set with respect to pandemic status. But this is just a rephrasing of the same data set, just showing pandemic, pre-pandemic versus pandemic to see what the sample sizes are. So it's decreasing in pre-pandemic and increasing in pandemic era. So the first model is this population model 1. It includes group time, group by time, and pandemic main effects. So we fit a diagnostic model to the data, and we find, yes, that this group time and group by time are not significant effects at all, really tiny chi-squares. So from backward elimination, we retain the group time, group by time, and pandemic. Not surprising. And a prognosis based upon this is that, well, there's no group by time by pandemic effect. So I can expect unbiased pooled data group by time effects. And there's no group by time or group by pandemic or group by time by pandemic effect. So I can expect unbiased pooled data group effects, and the same thing for the time effects. Now, as before, I said it's a good idea to look at the simple interaction effects. And so this does that, and all these p-values are pretty large, and that's not surprising. I mean, we know what the population model is, but in addition, up here, these chi-squares were really small, so it would be hard to imagine that any of the simple effects were significant, and they aren't. So there's no surprises there, but this is still an important thing, a step to take. So then, apologize a little bit for the fakeness of this and for the busyness of it, but I'll try to walk you through it so that you can look at it in a glance. So these are the simple group main effects, treatment versus control at baseline, at 6 months, at 12 months, and then this is the time average group difference. These are simple interaction effects. So this is just focused on the baseline to 6 month transition. Here's the baseline to 12 month. We've got within group time main effects, and then this is a group average time effect. After the E equals, the first number is the population parameter for that effect. And the second is at pre-pandemic. And after the comma is the population parameter for that effect in the pandemic era. So in this case, the expectations are identical for the pandemic and pre-pandemic and pandemic era. And so I've got them all listed in green. And so these are the parameter estimates from the Ancova model. And they're in green, that means they're unbiased. And these are the pandemic era estimates from the Ancova model, and they're all in green, and they're identical to their corresponding estimates during the pre-pandemic period, and that's what you would expect. And so there's there's really only one kind of general estimate, and that's, you could take either of these columns and that would represent it. Now, you can ask, I'm not showing the stratified analysis results here, but this relative efficiency is the relative efficiency of the Ancova estimate versus the stratified analysis estimate. So essentially, like at 12 months, if I fit an Ancova model, I effectively have a sample size that's 4 times larger than the stratified model for that effect. And it changes, but you can see that there's going to be a real efficiency gain in a situation like this versus doing stratified models. So you want to pool data if you can. So that was the first population model. This is a population model that includes a group by pandemic effect. And so now we have a different expectation for what the group effects will be during the pandemic era. They're not the same as these anymore, and so I'm showing them in purple. And so all of this is the same because there's no effects involving pandemic and time, but there is a group by pandemic effect. So I'm getting separate estimates of group effects by pandemic status that is just happening. This is just what the Ancova model is providing without me. I mean, I had to write those LS mean estimate statements, but they're super easy to write. And so I'm just getting this output. So you can see that there's an efficiency gain here and here. So essentially, I've got a hybrid of a pooled data. It's a pooled data analysis here, but up here, it's kind of like a stratified analysis, except that it's more efficient than a stratified analysis. So definitely, even if you've got like a group by pandemic interaction, like we do here, you still don't want to go to a stratified analysis necessarily. I say fit the stratified analysis because if you have a parameter estimate from a stratified analysis that's equivalent to the corresponding parameter estimate from any other kind of model you fit, then you know for sure that that is not biased. But there's no reason to suspect that any of this is biased. We've got fully crossed data with respect to pandemic and time, and we can fit the diagnostic model, and there should be no concerns. But in this case, you could either choose to, if you wanted to only focus on pandemic era effects, then you could just choose this column to report from. But you could also report the pandemic era group effects over here if you wanted to, or you could ignore them in your write-up. So this is one situation where I just wanted to compare the Ancova setup versus the Ancova setup. And again, I want to emphasize either approach could obtain the same results. But the way we set things up, because it's a group by pandemic interaction effect, what we asked Ancova to do was to give us to average across pandemic status. And that's what it's doing. So we have here the expectation pre-pandemic is 0, the expectation pandemic era is -1. So it's a weighted average, but in this case, it's about halfway between. It's about -0.5, and you can go down the line. So Ancova's doing what we asked it to do, but you may not want to report upon these pandemic averaged effects because in a lot of cases, they won't really be meaningful. It's possible that there's a kind of a group by pandemic interaction effect that is not substantial, so that the difference between the group effects across pandemic status aren't that big, so that reporting an average may not be a bad thing to do. That's a judgment call. But I just wanted to point that out. And so that's, this is the kind of reason why, based upon the way I set things up, I'm focused on Ancova results more than Ancova. So this is the third population model. There's a time by pandemic instead of a group by pandemic effect. So our time effects are going to differ. And we see efficiency gains everywhere except when you get into this time averaged or group average time effects, something most people don't report upon anyway. So I'll just move on from there. Same thing here. This is the all two-way interactions. So we've got even in this case, we've got group by time interaction effects that are equivalent across pandemic periods. So you might choose to report those in addition to pre-pandemic era group main effects. And then this is the worst case scenario in this sequence set where the population model has a three-way interaction. So essentially you're getting a stratified analysis here. You've got unbiased pre-pandemic estimates, unbiased pandemic era estimates, and the relative efficiencies are basically 1. So you're not doing anything better. There's no information other than complete stratification of the data set. Okay, so I had mentioned before that it's important to test the simple, if you find like a time by pandemic or a group by time by pandemic effect, or any kind of pandemic related effect, maybe if you had a RCT with 3 groups, then your group by pandemic effect would have more than 1 degree of freedom. You want to test the simple interaction effects involving pandemic for effects with 2 or more degrees of freedom. And so from population model 5, it was complex enough that both the time by pandemic and group by time by pandemic interaction effects had kind of 2 components. You could think of it as pandemic was modifying the baseline to 6-month effect, and pandemic was modifying the baseline to 12-month effect. So if we look at these, we see that there's a lot going on in that interaction. But what if it was less complicated than that? So I've made it a different modification of PM5 that includes a kind of a effect modification during the baseline to 6-month transition, but not from the baseline to 12-month transition. So here I see that the baseline to 6-month focused simple interaction is significant, but the baseline to 12-month is not. And then I fit both the Ancova and the Encoder models. And again, it's just the way I set things up. But in this case, Ancova setup doesn't do very well, except for this baseline to 12-month effect, where it's giving you the same estimate as the Ancova model for pre-pandemic, but the relative efficiency is better. So when you're dealing with interaction effects involving pandemic that have more than 1 degree of freedom overall, take this into consideration. You might be able to squeeze out a little bit more efficiency based upon your modeling approach. Okay, so we talked about sequence 1, 2, 3, 4. Now I just want to introduce the other sequences. So here, time and pandemic are not fully crossed. And so we've got no pandemic era baseline. Got this like missing cell right here in our cross classification. So with this, with S1, 2, 3, 4, our diagnostic model is only going to have 1 degree of freedom test for time by pandemic and group by time by pandemic, but we can still test them. The Ancova estimates were unbiased within each population model, but the time by pandemic and group by time by pandemic effects in the any of those effects in the population model would make it impossible to estimate pandemic era time effects beyond 6 and 12 months. Again, that's because pandemic era is only observed at 6 and 12 months. Oops. If you have a situation where you've got this sequence of observations, you could add pandemic era baseline, and then that would give you a full set of pandemic era estimates in your analysis. And in this case, Ancova did offer efficiency gains except for PM5. So here's just an example of an analysis of sequence 1, 2, 3 data. This includes a time by pandemic effect. And so here we've got the pre-pandemic estimates are all unbiased. We observe the pre-pandemic was observed at all time points with some efficiency gains, not across the board. In the pandemic era, we have equivalent effects for groups and group by time. Again, there's no group by pandemic or group by time by pandemic effect here. So that's what we expected with some efficiency gains. But for time effects, it gets, all we can estimate in the pandemic era are these 6-month to 12-month transitions. I also want to point out that for baseline group comparison and for the group by time effects that include baseline, there's the stratified analysis was unable to estimate these effects. But the Ancova model, is able to estimate those effects by virtue of the fact that they're equivalent to the pandemic era effects. So this is a case where the stratified analysis cannot estimate an effect, so to speak. It's estimated over here and they're equivalent, so I'm maybe making too much of a deal of it, but it's doing something that the stratified analysis could not. If you were focused on a stratified analysis, you would just get these 3 estimates here, for instance, and that estimate there. But they're the same as these, so it's all the same. Okay, 2, 3, 4 is the opposite of 1, 2, 3. Here we don't have a pre-pandemic 12-month assessment. And so you get the kind of the same situation, the same possibilities and results in a lot of ways, except it's flipped. Where here we got a full set of pre-pandemic estimates, an incomplete set of pandemic era. With 2, 3, 4, you would get a full set of pandemic era and a spotty set of pre-pandemic. And there's really no way to augment this design in most cases, because to do that, you would need to add some pre-pandemic, like 12-month assessment, which probably just isn't possible because you've run out of time to do that. Okay, looking at something more sparse is the 1 and 2 sequence. So now we don't have pandemic era at baseline or 6 months. And the diagnostic model has no information to test treatment by pandemic or time by pandemic or group by time by pandemic effects. They've got 0 DF. So it's the worst case. Well, worst case is PM5 with this. This is not the worst case. So here it gets pretty messy because pre-pandemic, we have unbiased estimates with PM5, so that this includes all up to 3-way interactions. It's essentially a stratified analysis for pre-pandemic, for the pandemic era, the Ancova model is producing some kind of biased results because it's just a bridge too far what you're asking it to do. It can give you a 12-month group comparison, and that's okay. But these other group effects at the pandemic era are messed up. And all of these time-related effects are identical to these effects because the model doesn't have any information to go on. So this is a case where the Ancova model will report pandemic era estimates that are not accurate because these are really pre-pandemic effects. They're not pandemic era effects. And we know that the population model includes time by pandemic and group by time by pandemic effects. So these are not truly those effects. We just don't have the data to estimate what those effects would be. So in this case, when it comes to pandemic era effects, I would just trust what you can estimate with a stratified analysis. But if you augment your design with a pandemic era baseline, then it would allow you to test this, where is it? Yeah, that one. It would allow you to test a group by time interaction effect across the baseline to 12-month transition. And so if that's important to you to have a group by time effect estimate in the pandemic era, then you could consider adding a pandemic era baseline to your study. If you're not interested in that, in the worst case scenario with this sequence 1-2, that is the worst case scenario being a population model like this one, you're going to end up with a stratified analysis of your pre-pandemic data, and that's all you're going to have. No efficiency gains. But that's assuming if you have a population model 5, you never know what you have, but until you do analysis. But if you added, oh no, I should just say that without adding a pre-pandemic baseline, the Ancova approach for this kind of data layout will have efficiency gains over stratified analyses if there's no group by pandemic effect that's operating in the population. That's not shown up here, but it's just a point. Okay. With 3-4, it's kind of the same as 1-2, so I'm just going to skip over it. But I do want to focus on this 2-3. This is like the worst of the worst. So here we've got completely pre-pandemic base, completely pandemic era 12-month, and a mix at 6 months. Again, the baseline to 12-month comparison is completely confounded with pandemic status. So it's a tough case. If we assume the worst of the worst and that we've got a population model that includes the 3-way interaction effect, and we've got sequences 2 and 3 only, then we just get this big kind of hodgepodge of basically it's a stratified analysis, and you know what you can get pre-pandemic kind of goes up to 6 months, and what you can obtain for the pandemic era that's that's at least not biased has to do with 6 and 12 months only. So it's kind of a mess in terms of, unless you're happy with the stratified analysis that only goes to 6 months pre-pandemic and a simple group comparison at say 12 months and 6 months during the pandemic era. I mean, you could do it, but you're going to be losing a lot of power depending upon what your balance of sample size is across pandemic eras. So again, you'll get some efficiency gains if there are no group by pandemic effects in the population. But again, you could consider, and I would especially consider in this case, augmenting your design with pandemic era baseline observations. It'll give you a full set of pandemic era estimates, which is going to be a big improvement over this circumstance. And it will give you some gains in relative efficiency, except in the worst case, where the population has a three-way interaction effect operating. So, final summary. The Encova setup that I described, it offers a convenient hybrid approach that produces both pooled data and stratified estimates as appropriate. It kind of just automatically does it, and can have efficiency gains over stratified analyses. So it's almost a one-stop shop. And so, I made some summaries here about, depending on what the diagnostic model finds, the kinds of models that you could fit in response to that. I think it's, not much to go into here. Just a reminder about the complex interaction effects. There may be different ways to estimate what the simple effects are that are more efficient than others. I think it's always a good idea to compare estimates across stratified and ANOVA and Ancova. I mean, if they're the same, then there's certainly no concern. If they're different, then there is a concern. If the stratified analysis is unable to produce an estimate that the Ancova model can, it doesn't mean there's a problem with the Ancova or ANOVA model. It's just requires further scrutiny on your part. And the problem really becomes when the cross-tabulation of pandemic and time gets really sparse. Then you may be asking the model to do too much. But in our, in the simulation with pattern 1, 2, 3 or 2, 3, 4, it wasn't a problem. It was only when we started getting into sequences that were sequence sets that were more sparse than that. So my advice is to plan ahead for your worst and best case scenarios. I mean, look at your projections for what proportion of your sample will fit into the various patterns or sequences of pandemic status with their longitudinal observations, and get a statistician and start conducting some simulations to try to understand what the effects are going to be and what the power analysis might look like under different population models. Whether or not you should, if it's an option, whether or not you should think about enhancing or augmenting your design by adding at least a pandemic era baseline. It's a possibility, but the upside of this is that there's really a lot that can be done and a lot of efficiency gains that can be obtained. And it, we will see. We have yet to understand how prevalent it will be for there to be like group by time by pandemic interactions. Maybe they're kind of few and far between, and so that a lot of studies at least will be able to report pooled data, group by time interaction effects, for example. So we'll see. Maybe it's not going to be as big of a problem. If all we have are pandemic main effects, then there's no problem. You just include pandemic in your model and model as you would normally, and everything will be taken care of. So time will tell, and that's it.
Thank you, Steve. Thank you very much. That you made this setup very clear and almost simple, but this is very complicated. do you have data that you actually analyzed using some of these methods?
Just simulated data.
Yeah, so it's
Yeah.
simulated data. No, you don't have.
No.
Yeah.
Yeah. So yeah, and I mean I could share the code. It's a big giant program, and it's scary, and it's got a bunch of
sure, that will be
macros in
great.
it, so it's not easily. If you modify it, you might break it, but because it just deals with a lot of contingencies, but it was instructive.
Anyway, I don't know. Oh, there are a lot of things that showed up in the chat box now.
I'm gonna stop sharing so I can see the chat.
Yeah, people are asking, and the slides will be available, and the recording will be available. Just to let people know. So, do you want to go over the chat box, Steve? Now.
Yeah, sure. So,
okay.
Thank you, Jesse. Yeah, Torres mentioned on slide 9 that the group by time interaction should be in there. I thought I caught that verbally, but yeah, that needs to be corrected. That should be in there. The diagnostic model should be the saturated model all the way up to the three-way interaction.
Right.
Yeah, Jesse asked about missing data. It just kind of depends on the study, and I can only say anecdotally, a lot of people are reporting that their retention is better, especially if it's remote data collection, because a lot of people, not everyone, but a lot of people have less to do while they're sheltering in place. But obviously, and in some cases, even where a kind of an in-person clinic visit was required. It used to be that studies would often have the patient or the participant come to UCSF to have their blood drawn or whatever. But now that hasn't been, or at least it wasn't originally acceptable. And so studies that wanted to continue with this kind of required intervention or visit would maybe hire private nurses to go to the participants' homes and collect the specimens or the biometric data or whatever it was. And so that just made it more convenient for the participants. And so they actually had some gains there. But certainly there's going to be other situations where things are worse in the pandemic era with respect to participation rate and retention and all of that. I completely ignored missing data in this talk. Tor's talk last month focused a lot on missing data and strategies for dealing with it. So I'd look at his slides, which should be available on the website. Yeah, I mean, again, for outcomes missing due to pandemic, I would look at Tor's slides. I mean, essentially, you need to have a principled approach to dealing with missing data, and that could be a likelihood-based regression model and/or multiple imputation or other kinds of things along those lines. So, the best you can do is a principled approach, and you can have sensitivity analyses that kind of vary the assumptions of the imputation model, et cetera, to see how robust some of those assumptions might be. So, but I would just refer you to Tor slides because it's pretty standard imputation approach, just with the extra accommodation of pandemic status in there. So this is the Ancova. The Ancova approach might be safer than the Ancova approach. So I think so it gets a little tricky deciding. So in these situations like this S12 or S23, you can get some Ancova estimates that are biased. And so I haven't figured out exactly what the empirical circumstances are that will lead to that breakdown, but it definitely has to do with how sparse the pandemic by time cross-tabulation is. In this simulation, where there was only one missing cell on that cross-tabulation, there weren't any problems. In cases where there was two missing cells in that cross-tabulation, there were problems. And so if you are concerned about that, my advice would be to perform simulations to try to understand whether or not some of the Ancova effects might be biased given your particular data setup. But definitely the safe harbor is if you do a stratified analysis and you get an effect estimate and it's basically equivalent to what you got from Ancova, then those Ancova estimates for sure are going to be okay. At least as okay as they can be. There could always be bias because of missing data and things like that, but those are going to be okay. And yes, the slides will be on the CAPS website. Okay, I think that's it.
Okay, thank you, everybody. You will be getting email that you should reply to with the evaluation of the presentation. And this is important for our funding institution. Thank you very much and stay safe.